I'm Judith Ank. I'm the president of the organization Beyond Plastics and a professor at Bennington College in Vermont. We are so happy to have you join us this evening for this educational webinar with some amazing speakers. Thank you for giving up your beautiful Thursday evening. Beyond Plastics is a nationwide project based at Bennington College. We work on opposing plastic pollution everywhere. We work on the production, use, and disposal of plastics. We work with grassroots advocates around the country, and we support public policies at the mostly state and local level that will reduce plastic pollution. We all know by your presence tonight that there is heightened public interest in the issue of plastics and health. When I first organized Beyond Plastics, I just wrapped up my tenure at the Environmental Protection Agency, where I served as regional administrator appointed by President Obama. And I had a lot of time to think about what to do next and decided I wanted to work on plastics because there was an unmet need. And I love big, audacious, difficult problems that we want to solve together and involving people at the grassroots level. And quite honestly, it was the issue of plastics in the ocean that really grabbed my attention, the fact that we're turning our ocean into a watery landfill. And it was the issue of climate change, because plastics are made from fracked gas, a type of fossil fuel, and also 16,000 different chemicals. Plastics is an environmental justice issue because production is mostly in the Gulf states and Appalachia, causing tremendous public health damage to our brothers and sisters living in Louisiana, Texas, and Appalachia. I love recycling, but plastics recycling doesn't work, coming in at a very low recycling rate of 5 to 6%. And now we have the health concerns with microplastics and nanoplastics. We know that due to important scientific papers, microplastics have been found in our brain, in our heart arteries, in our lungs, in our kidneys, in our blood, in human placenta, both the fetal side and the maternal side, breast milk, and testicles, just to name a few. And so we thought it would be good to invite 561 of our closest friends this evening to hear from a couple really wonderful technical experts. And I just want to announce, those of you that live or work in New York State, we're going to have a little overtime at the end of this program to talk about a really important bill that is pending in the New York State legislature to reduce plastics, which has really gotten the attention of New York State lawmakers. So if you want to stay for overtime with us, there'll be snacks, drinks will be flowing in your own homes. And we want to talk to you about that bill that's pending in New York. So let me please begin by introducing our friend and colleague, Dr. Kathy Nolan. Dr. Kathy Nolan is a medical doctor. She's a pediatrician, and she is with Physicians for Social Responsibility, New York State. And PSR is co-sponsoring this webinar tonight. Dr. Nolan is a pediatrician and researcher who's focused her work on addressing toxic contaminants in our environment that cause or risk harm to humans. Dr. Nolan is on the board of Concerned Health Professionals of New York, an extraordinarily important group of medical experts. And she serves as the president of the New York chapter of Physicians for Social Responsibility and is author and editor of the very important document that we'll drop in the chat. It's called the Compendium of Scientific, Medical, and Media Findings Demonstrating Risks and Harms of Fracking and Associated Gas and Oil Infrastructure. Historically, plastics were made from chemicals and oil. Now it's chemicals and a waste product from fracking. So if you want to work on plastics, you've got to get really smart on the fracking issue. And Dr. Nolan has made it very easy in terms of creating this document. So Dr. Nolan.
Thank you very much. Yes, my colleagues and I put together the compendium of pulling together papers on the risks and harms of fracking and fracking infrastructure precisely because we thought that the public needed to have this information. And approximately 10 years ago, when we started, there were only a few dozen papers that actually looked at direct health impacts of fracking the gas and oil and every step along the way. Now we have over 2,000 entries. And we've looked at every phase of the fracking activity. Tonight, we're going to be looking at plastics. And we don't often think of that as the last phase of fracking activity, but it is that and burning gas inside our homes. And part of the work of concerned health professionals and physicians for social responsibility is looking at keeping gas and oil combustion out of our homes in terms of appliances, including stoves in the kitchen. But where we really see plastics entering our lives, often in ways that we don't notice, is when we are using plastic products, which are a waste product. And we might think that it would be harmless to use that product, but we shouldn't necessarily assume that because every phase of gas extraction, we know that there are harms from chemicals that we put in, but also from chemicals and components that come up out of a well bore with fracked gas and oil. So those continue through the transportation, the storage, and the disposal of waste, but also the hidden waste that is in our plastic products. I'm very much looking forward to these presentations tonight because if we're going to have the cultural change we need in relationship to plastic, which I think of as being like the cultural changes around seatbelts and cigarettes, those were based on having data about how much good could be done and how many lives could be saved by making changes at the societal level. So let's uh, let's dig in for some direct evidence about the impacts of plastics in our bodies, and then also think a little bit broadly about what that means that we should do in terms of changing the way we live.
Thank you, Dr. Nolan. And now we're really excited to welcome Vanessa Fadgens-Turner, who will also welcome you all this evening. Vanessa is the Executive Director of Environmental Advocates of New York, the state's premier environmental advocacy group in Albany. Before joining Environmental Advocates as the Executive Director, Vanessa worked on sustainable development with a focus on climate change. She also had this whole other career as a producer for seasons 1 and 2 of National Geographic's Emmy Award-winning documentary, Years of Living Dangerously, which you should all watch this weekend if you haven't. Vanessa is a term member of the Council on Foreign Relations. She is a sister and an ally and just a great partner working on environmental issues in Albany. Welcome, Vanessa.
Thank you so much, Judith and Dr. Nolan. It's wonderful to be with all of you here tonight. And for those of you joining us from outside of New York State, as Judith mentioned, EA, Environmental Advocates, is New York's one of its largest and its oldest environmental advocacy groups focused on our state level policy here. We've been around for over 50 years and pushing for laws that protect clean air, safe water, public health, and are very proud to be partnering with Beyond Plastics and Judith this session and always. As Judith mentioned, before stepping into this role, I spent several years working on various aspects of plastic pollution and its byproducts in the ocean conservation world, and later with the United Nations in sub-Saharan Africa. There, plastic waste is both a symptom and a stressor of broader environmental justice issues. And what's heartbreaking and infuriating is that we're now seeing these harms spreading with more cognizance about the ramifications here at home. Microplastics showing up in our water, our air, and all throughout our bodies. But what I'm here to sort of help elevate is that in New York, we have the power to do something about this. This is why EA is so proud to sponsor and partner on tonight's conversation, and always to support one of our favorite partners here, Beyond Plastics, in the fight to pass the Packaging Reduction and Recycling Infrastructure Act, or as we say, PREA, which I know Judith will speak about further at the end of this. And I always want to give a special shout out to Judith and Beyond Plastics, who are trailblazers on this issue. And I'm proud to say Judith is one of my predecessors at Environmental Advocates for my role. She and Beyond Plastics are instrumental in elevating this issue nationally, but especially here in New York, and their leadership has really helped build the momentum that we have today. So I'm really proud to be here. This isn't just a global crisis to discuss, it's a local fight. And in New York, it's a fight we can win. So thanks for all joining.
Thank you, Vanessa. We are members of the Mutual Admiration Society. So you're probably thinking, how is tonight going to unfold? Here's the game plan. We are going to hear from two amazing medical experts who are going to make us smarter and so much more informed than you were just 15 minutes ago. We're keeping the chat off until after they speak, because I find it very distracting when I'm speaking because I keep looking at the chat questions. So we're going to spare you that experience, which I find traumatic. So we'll hear from our two experts, then we're going to open the chat. You can type in questions that I will pose to our experts. And then a surprise, when the webinar ends, we're going to go into overtime. And if you are interested in hearing the latest about the New York Packaging Reduction and Recycling Infrastructure Act, you're welcome to stay on. I know this is a surprise. Surprises are good, but there are only 3 weeks left in the state legislative session. So if you live or work in New York, or if you have a kid or a niece or a nephew in New York and you want to hear more about this bill, just stay with us and we'll go into the details and take any questions. So it is my great, great honor to introduce our first speaker tonight, Dr. Sanjay Rajagopalan, who is Chief of Cardiovascular Medicine and Chief Academic and Scientific Officer at University Hospital's Harrington Heart and Vascular Institute, and Director of the Case Cardiovascular Research Institute at Case Western Reserve School of Medicine in Cleveland, Ohio. Doctor is among an elite group of physician scientists whose work has really helped transform the global perceptions and understanding of the impact of environmental risk factors on cardiovascular diseases. He won the 2021 Distinguished Scientist Award from the American College of Cardiology. He has published over 300, oh my goodness, over 300 original peer-reviewed research publications in journals such as the Journal of American Medical Association, New England Journal of Medicine, Circulation, that sounds like a fun journal to read, Journal of Clinical Investigations, and Circulation Research, in addition to more than 300 invited editorials and reviews. So in short, he's a really big deal. And we are so impressed with his papers and his work, and I'm delighted to welcome you this evening.
Judy, you're very kind. It's really an honor and a privilege to be joining this very interesting webinar with you today, and to be joined by Kathy and Vanessa. And thank you, Kathy, for all the work that you do, and Vanessa. It really is a very important problem, and clearly it's a privilege for me to be joining people who are actually doing the work and trying to publicize this issue. So as you might have surmised, I'm a cardiologist, and one could ask, why is a cardiologist interested in pollution or plastics? And it happened many years ago when I got interested in air pollution, nearly 20 years ago. And it turns out that a number of pollutants, including plastics, are sort of similar in the sense that many of the constituents of these as you mentioned, extraneous materials, which is produced primarily through fossil fuels and manufactured for a variety of different reasons, has an annoying, a rather dangerous side to it. They get into your body, they penetrate the organs in your body, and in many ways, they act as a biological intruder. And just as this story of air pollution evolved, and as Vanessa mentioned and Kathy alluded to, we need evidence to demonstrate that these things are actually harming and producing health effects before we can request, or rather, envision a cultural change, which is really what's required, because these plastics in particular are such a pervasive problem, and in some ways very similar to air pollution. And we sort of consume plastics, we inhale plastics, we absorb them through our skin, we ingest them through our GI tract unknowingly. Many a time, most of us are sort of innocent consumers of these materials, because many a time you don't have any choice. Now, being a physician, I know the incredible impact of plastics, right, in the medical industry, where they are really life-saving materials in so many ways. But then again, there's the other dark side to it, where they've permeated the environment that we live in to such an extraordinary degree that it's almost inescapable not to encounter plastics in every facet of your life. And as I mentioned, they have this really annoying aspect of getting into your body. So I'm not going to go through the scope of the problem. I mean, you're all very seasoned and well-educated consumers of news and materials, so I'm not going to restate all the statistics that you already know, so I'm not going to really talk about that. I guess what I'd like to do is to spend the next 8 to 10 minutes discussing some of the, I guess, a top-line summary, if you will, of what we do know about microplastics and nanoplastics and how they affect the cardiovascular system. And Marcus, who's done some really good work in terms of the neurotoxicity and materials that make their way into the central nervous system, can really talk about some of the neurotoxic effects and what we do know from experimental models in the human condition. So there's no question that the body of evidence to date seems to support a strong evidence that microplastics and nanoplastics could be potential risk factors for the development of cardiovascular disease, right? And it's not just the plastics or the degradation products of plastics, which is principally how we view them, when microplastics are actually derived from macroplastics, where they are sheared off and degraded either by mechanical or chemical means. And these fragments produced as a consequence are widely distributed throughout the biosphere, right? And they're detectable in just about everything that you eat. I just saw a paper in Nature published, I think, a couple of weeks ago, showing a very facile route by which they're absorbed through leaves of a plant, right? This is a very high-profile paper that got a lot of publicity. And in fact, it might be more efficient in absorbing microplastics than even the roots, which has traditionally been the thought process of how nanoplastics are absorbed into plants, right? And that's a very, talk about a Trojan horse, right? So essentially you're eating plants, and these materials sneak into you when you're eating your salad. Not a very comforting thought, but that's the scope of the problem. And they're just about present in. And if you do experiments on looking at bottled water, obviously, I think most of us in the audience and people listening in know that they are pervasive, in water as well as in food components, because plastics are almost ubiquitously used to store foods, right? So I think let's start with the evidence so far. I think there's been a lot of in vitro evidence where microplastics and nanoplastics. And I think we'll sort of also talk a little bit later on, perhaps in conjunction with Marcus, about the plastic-associated chemicals. And as Kathy mentioned, there are more than 16,000 chemicals. And, the sad part of chemicals is that to date, it keeps changing. There are more than, I think, 400,000 manufactured chemicals that we know of which we know something about them from a health perspective of less than 1,000. So it's just a bewildering array of chemicals. And these chemicals are obviously used in conjunction with plastics in order to endow them with a variety of different properties, right? Which could be things like plasticizers or phthalates that make plastics soft, etc. So we won't get into that. So there's the other aspect of plastic-associated chemicals, which also have independent health effects. And increasingly, there's a recognition that they might induce a number of health risk factors, cardiovascular risk factors like hypertension, for instance, or insulin resistance and diabetes. And the literature for some of these chemicals is actually pretty good. But let's step back a little bit and look at in vitro models clearly show that these microplastics and nanoplastics are viewed by cells as foreign material. And there's a well-established, in fact, a robust body of literature pertaining to how cells perceive foreign material. There are legions of foreign materials, unfortunately, lots of foreign materials, I shouldn't say legions, lots of foreign materials that make their way inside the body. A good example of this is cholesterol. So, I'm a cardiologist, very interested in how atherosclerosis, which is a process by which, your arteries harden and you get a heart attack, right? You get plaques that develop within your circulatory system. The plaques might rupture, and all of a sudden a clot forms on top of it, and you get a catastrophic heart attack. So the first thing to know, I think this is sort of a little bit of education here, is that heart attacks don't develop gradually by progressive narrowing of the artery, as is a popular conception. It happens abruptly, dramatically, because of a little rent in the arterial wall, much akin to a crater that might develop on a road, and all of a sudden there's a huge clot that forms on top of it. So cholesterol deposition into the arteries is what causes plaque formation. And these are oxidized cholesterol or oxidized lipids. And when these lipids are brought into and deposited into the arterial wall, they create what we call an inflammatory reaction, right? So they set off an inflammatory cascade, which results in a number of other cells that get into it, just akin to a wound that's trying to heal, where you have white cells that move into the area and try to clear up the cholesterol. In an attempt really to seal off the inflammation, but when you do that, you sort of get this hyper-inflammatory response in the plaque. And cholesterol crystals are the best studied examples, both in experimental animal models and humans, that elicit an inflammatory response. Then there's also things like urate crystals and gout. There's obviously a lot of data with silica. We get silicosis when you breathe silica. There's asbestos, there's silica. All these are foreign materials. And what do? I mean, foreign materials are biological intruders. They have no business being there, right? So at a very fundamental level, you have to ask yourself, I mean, this is not normal. When you find microplastics and nanoplastics all over your body, well, this isn't normal, right? So you could sort of pretend it's going to be innocent, but I mean, there's never been an example of an innocent bystander in the body, just happens to be there. Okay, now you don't have to believe me. You could say, look, we don't know that. You have no evidence to show that. And let's explore the evidence. So the first concept is the concept of them being biological intruders that are in tissues, and they should not be there. And they induce an inflammatory response. They also cause what we call oxidative stress mechanisms. They induce the generation of radical species. And when that happens, these radicals have a nasty way of involving just about everybody else in the vicinity. It's like a bad element, right? Once a bad element gets in, the entire neighborhood goes to bust. So that's really what happens. So at a very fundamental level, this is what happens in cells. So cells are very happy, and we can talk about cardiovascular cells like endothelial cells or macrophages. Macrophages are professional garbage collectors, right? And these macrophages are good guys. They're present in your lungs. They're also present in your arterial system. They move into an area of inflammation and they try to ingest the particle. Now, when they try to ingest microplastics and nanoplastics, it turns out that these ingestion that happens is not so innocuous. And the macrophages are not very happy. And what they try to do is they try to digest the microplastics out, but in doing so, they actually release a lot of enzymes and cytokines into the area that they are there. And they sort of have this collateral damage effect to cells as well as plaque. So this has been well studied in experimental animal models. And again, as I mentioned, with all routes of entry, right? You can inhale. There have been experiments in animal models with inhalation. And again, a lot of these, for those, obviously we're talking to a sophisticated audience, there's a lot of, you can criticize a lot of these. You could say, well, some of these materials are really not relevant to human consumption. You can't inhale this dose of polystyrenes, when there are lots of people that'll throw up their hands when you put up a paper and with an experimental animal model and talk about the size of these particles as being not relevant to human exposures. And those are all fair points. I mean, animal models are animal models. I mean, they give you some pathways and cues, but they certainly, I think, provide a body of evidence in terms of initial pathways that you can investigate with more sophisticated approaches. So, and what we find is really, regardless of the type of nanoplastics you use, and regardless of what type of route of delivery, obviously inhalational routes are pretty toxic. And most of the studies have been done at relatively high exposure levels. So for that reason, you see a lot of lung toxicity. You also see a lot of systemic penetration of these particles into the brain, into various parts of the body as a consequence. But GI absorption, where animals are given concentrations of a variety of different nanoparticles and microplastic particles, have also shown the same thing. They show that they get absorbed. They get into just about every organ system. And what you start to see is the smoldering signs of inflammation. Now, when they've been given to experimental animal models with disease models, in other words, there are animal models that mimic the human condition where they have this condition called atherosclerosis, right? And what we find is when you do experiments with prone animals, prone meaning that these animals have predisposition for developing plaques, just like the human condition, they certainly make plaque development worse and they worsen atherosclerosis. Then there's the human experiments. There's an emerging body of literature showing accumulation of microplastics and nanoplastics in just about every portion of your body. It's in the placenta, it's in the brain, it's in the lungs, it's in the heart, it's in the arteries, it's in the liver, it's in the GI system clearly, the spleen. No organ is spared, really. It's just about in every system. And there have been studies that have tried to estimate the exposure. In other words, how much of plastics are you actually consuming, right? Each human being that's consuming. And there's a wide variation of this because there's a lot of variability. Obviously, these are not carefully crafted measurement of exactly what human beings are consuming. So these are all estimates, right? You make estimates by making assumptions about things. So there's a wide variation of this, but the conservative estimates are anywhere from 5 grams a year. The previous estimates were wildly, out of whack, and they got criticized. But more conservatively, I would say anywhere from 5 grams to as much as 50 or 100 grams even. You can look those papers up. But nevertheless, and that's in the GI route, total exposure, a variety of different routes. Now, the cardiovascular system, as I mentioned, you find them in a lot of tissue. I'll profile one particular study that we did, and this is really the work done by colleagues in Italy, from Milano and Naples, where they actually followed. The experiment goes something like this. So it's extraordinarily difficult to get tissues from humans, right? I mean, I shouldn't say this, but you have to wait for people to die to get tissues. Sometimes you can get biopsies of patients from skin biopsies or, visible tissues, but it's hard to get cardiovascular tissues. So how do they do that? Well, turns out that patients undergo a procedure referred to as carotid endarterectomy. Carotid endarterectomy is a procedure where they literally make a small incision in your neck, and this is to treat a condition called carotid stenosis, which is a blockage in your carotid arteries. The carotid arteries are 2 arteries that go into your brain and supply blood flow to your brain, and when you develop a blockage, you're at high risk for stroke. So carotid endarterectomy is a procedure where, typically a surgeon, it can be done with the balloon procedure as well, but when a surgeon does it, he does a little incision, and then he goes in, he opens up the skin, he exposes the carotid artery and literally scoops out the plaque inside the artery and sews it back again. So then you could take that plaque tissue and then analyze it. And I'm sure Marcus will talk about his fantastic technique that he uses at New Mexico. And this is a technique called pyrolysis, GC gas chromatography, GC mass spec. Very sensitive technique. This allows you actually to take a fingerprint of the concentration of microplastics. So you can actually derive a concentration. And that's exactly what they did. They found the concentration of microplastics within the arterial wall in the carotid plaques. And they found two types of plastics. These are my colleagues. I was part of the publication, but really they did the work. So two types of plastics. One is polyethylene, and the other one was PVC. Polyethylene out of, I think they recruited about 257 patients who are undergoing. So this is a big deal. I mean, getting 257 patients recruited and getting their plaques out and examining it, and then following them over the next 3 years to see whether they actually had incidents of stroke, heart attacks, and death, right? That's another outcome. So as cardiologists, we want to do all we can to prevent a heart attack or stroke, which happens through a very similar process, or cardiovascular death, which is the most common cause of death, period. So that's what they did. They followed these patients for 3 years. And out of the 257 patients, 150 had evidence of polyethylene in their plaque, so more than 60%. And a small minority of them, I think in the 30s, had evidence of PVC. And when they looked at the histology, when they actually looked at this under the microscope, they also found smoldering evidence of inflammation. And the particles looked quite nasty. These are jagged particles with sharp edges, very similar to cholesterol. Cholesterol also has this attribute where it's very, uneven and jagged. And the shape and morphology of these particles also have implications for the types of reactions they set off, right? These are not smooth, particles. So the long and short of it was that this was perhaps the largest study amongst others that demonstrated the evidence. Now, and I'll say this and stop because I probably am very close to my, 10 minutes allotted to me, and I want to provide enough time for others. I would say that this is the type of evidence that we have. And, people who are naysayers or sort of critics would say, well, that doesn't prove, just the presence doesn't prove causation. You haven't shown me causation. You're not showing me that this is exactly what's causing it. There are lots of examples of things that happen to lurk around in a tissue and it does nothing. And this goes back to what I said at the beginning, which is the biological intruder concept, right? So when a particle is no business to be there, you can pretend, you could say, well, this has no effects, but to the best of our knowledge, in every situation that we've encountered this type of thing, there's always a consequence. And I can't think of a situation where there's not been a consequence. So I'll stop there, but thank you.
Thank you. Thank you, Doctor. That was excellent. And we all read that New England Journal of Medicine article with great interest, and we'll share it with everyone on this webinar, and we'll drop it in the chat. And I will now use the phrase from you, biological intruders that should not be there. That is, that's etched in my brain, which hopefully does not have too much microplastic in it, but it probably does. So we are now really grateful to have Dr. Marcus Garcia speak. Dr. Garcia is a postdoctoral fellow at the University of Mexico's College of Pharmacy, Department of Pharmaceutical Sciences, specializing in toxicology and pharmacology. Dr. Garcia is dedicated to advancing our understanding of microplastics and their implications for human health, reproduction, development, and of course, on our shared environment. Dr. Garcia has made significant contributions to the field of toxicology and pharmacology through his pursuit of excellence in both research and teaching, and has currently developed a novel technique in the quantification of microplastics in biological tissues. His research portfolio spans various groundbreaking projects, and I don't say that lightly. They really are groundbreaking projects on microplastics in the human placenta and other biological tissues. Please join me in welcoming Dr. Marcus Garcia.
Awesome. Well, thank you very much for that introduction, Judith, and to everybody here. Thank you for your time and taking away some time from your families this evening to spend some time with us and discuss. And I think with the discussion that we're having, I think Sanjay, it's a perfect segue from a lot of the points that he discussed, and a lot of the areas that I'll be discussing this evening as well. So I'm just going to go ahead and pull up a little presentation that I have put together. But really, just to kind of get started, I know everybody on this call is very well educated around plastics, so I won't take so much time around the background of it all. But I mean, one thing that just to help put things into perspective whenever we're talking about plastic production as well as the formation of micro and nanoplastics. I mean, we all know that plastics production really started back in the 1950s, and it has grown exponentially since that point to where we're actually seeing a doubling of production every 10 to 15 years. And what that also means is that we're seeing that same doubling effect of microplastics in our environment as well. So really the things to kind of think about is even if we were to stop our plastic productions today, just because of how much is ubiquitous in the environment, we still see that continual growth of microplastics and nanoplastics in the environment. So really going beyond where we're currently at to be able to think about like what are the next steps in plastic or removing plastics or trying to figure out policies around that as well. So I think Judith also brought up a really good point about this is whenever we talk about microplastics in various ecosystems, I mean, we all know about the gyres that are throughout the Pacific Ocean and the eastern and western garbage patches that are really just free floating within the Pacific Ocean as well. And that is one thing that is of the utmost concern, and really where microplastics research first started. But I also like to show this to give some reference just because I had an opportunity in this whole microplastics research journey of mine to go work with the Hawaii Wildlife Fund and remove a lot of this plastic material from some of their remote areas and really see how a lot of that is affecting the ecosystem. And while I was there, one thing that really struck home for me is if we can all see this picture here. I know all of you scientists out there who would be able to identify what this is right away, but this is actually a pipette tip that we use every day in the lab. And just to be able to find that in such a remote area, it really makes me feel like as a scientist, we are also part of this issue, and there's a lot of ways that we need to even work to improve this as well. So I always like to put that into perspective. But also too, especially coming from the University of New Mexico, we are a landlocked state. And when we first started looking at microplastics and potential exposure routes, and I think Sanjay hit a lot of these points in his discussion is where are we getting a lot of our exposure from these things? So, I mean, there's not an ocean source very close to New Mexico as it is. So when it went into the investigation, a lot of our exposures is actually coming from our plastics that end up in our landfills. So for example, you have those plastics that end up in a landfill, they start to degrade from being exposed to sunlight as well as that mechanical degradation as they're being packed down. Well, over time, what ends up happening is as those plastics break down, they leach into our soils as well as into our groundwater. And then through the soils, those get transported into our plants and then into our agriculture. And ultimately from there, those also end up into the food chain. So, I mean, we used our groundwater to irrigate crops, and then we have that growth. I mean, we see our livestock consume those plant materials. And then ultimately, because of their accumulation of microplastics, we consume the livestock material. And that's one of the ways in which we're getting majority of our exposure. And as Sanjay did bring up is having that transport through the GI tract. So really something to consider whenever we're thinking about all of this. But to touch base on some of the reasons why we really wanted to take the next steps in developing techniques to better identify microplastics. Prior to the technology that we've developed and been using is a lot of the stuff around microplastics identification was really from a visualization standpoint. So one of the things that had been used in the past is a lot of microscopy, visualization, or primary analytical techniques such as Raman spectroscopy, as well as Fourier transform infrared spectroscopy, which are great techniques to be able to see. However, they are limited into how much of the materials we can actually look at, just because usually with Raman and FTIR, we're limited to only seeing particles that are larger than 5 microns in size. And whenever we think about microplastics, I mean, we're thinking even now looking at nanoplastics is we're now in the one micron or smaller realm to really be detecting these things, as well as being able to figure out ways in where we can detect plastics from the biological tissue and be able to decipher those from potential carbohydrates as well as lipids. So that ultimately led us to the development of the technique that we're using. So for example, and what I'll be talking about is within the brain itself. So we're able to take biological tissues, and this is fresh tissue or formalin fixed tissue. And roughly we need about 500 milligrams of that. And we put in a very, very basic solution, so a 10% potassium hydroxide solution. And we allow that to incubate for 72 hours at 60 degrees C. Then what that ends up doing is it breaks down the biological matrix completely and only allows so pretty much free floating within that is going to be those micro and nanoplastics. Well, then what we're going to do is we use ultracentrifugation, so really fast speeds, so roughly around 100,000 Gs over a 4-hour period, to be able to pelletize the plastics and remove those from that biological matrix that's remaining. And then we do some washes to clean the pellet off from that. But then ultimately, we're going to put that pellet into a pyrolysis cup, and then we do the combustion technique, where once that pellet hits the pyrolysis unit, it combusts at 600 degrees centigrade. So pretty much turns everything into a gaseous phase. And as Sanjay mentioned, it really creates that fingerprinting to be able to look at the different ions that are associated with the different types of polymers. And then we have a known calibration standard curve that we use of these microplastics of interest to be able to identify and give us an adequate dose of what that is. So just to kind of give you an idea of the types of polymers that we can look at, polyethylene, polypropylene. We can also look at polystyrene, nylons, polyethylene terephthalate. So a lot of very common plastics that we see in a lot of food products, as well as everyday products that we're using throughout our household and other areas as well. So that method had been used for a number of different publications. So the first publication for that, and really where the method development came, was for our placenta paper that was published in early 2024 in Toxicological Sciences. But really amplifying that technique, this is where our study looking at microplastics across organ systems and specifically the brain came into play. So at the University of New Mexico, we also have access to our New Mexico Office of the Medical Investigator. So we have our collaborators listed here. And within that, we were able to collect samples from human decedents, specifically liver, kidney, and brain. And we also wanted to frame this around looking at over an 8-year period to kind of model that 10 to 15 year increase in plastics in the environment to see if we were seeing any differences. And ultimately, some of the data that we had specifically in the liver and the kidney, we did see that there was an increase in plastics accumulation over that 8-year period. But one thing that was interesting was in the liver itself, there really wasn't any significance. And as you see some of the images, there's a lot of things that are really at play here. So our body has a natural way of, really trying to get rid of these toxicants out of our body. And in a certain ways, they're able to. And I think the kidney is a good example of that. However, because we have a lot more plastics in the environment in 2024 versus what we're having in 2018, that's also part of the reason why we're seeing such a higher accumulation. And to give the liver an example for as one of the ideas to look at. But also one thing we always like to do is make sure that we validate all of the stuff that we are doing. So we also collaborated with Oklahoma State University on this project. And if you see here within our brain samples, we also sent them some tissue, and they did the analysis in their own lab using their own pyrolysis gas chromatography mass spec system. And what they were able to show is that their data actually matched exactly what we were seeing as well too. So that was one of the validations that we performed. Within this study, something that we also had access to was we had about 12 dementia cases that we were able to analyze. So 3 coming from vascular dementia, 6 from Alzheimer's dementia, and then 3 from other types of dementias. And within that, one thing that was interesting is if you see within our collection years, everything here in red was our cohort from the University of New Mexico. But within this study and within the review process, one of the questions that was brought up was, well, what about everywhere else? How is that going to play a role? So with that, we worked with Andy West at Duke, where he was able to provide samples from the North Carolina area, Maryland, Massachusetts area as well too. And that was from about the late 90s all the way up to around 2013. And if we can see here, we see that same increase in trend. But going back to our dementia cases, one thing that was very interesting was that we saw that there was about a 10-fold increase in the amount of plastics that were present in our dementia cases. Now, we are still at the point where we're saying this is more of a correlation, not a causation. But just with this finding alone, it's really amplified our studies and our interest to be able to continue to answer this question, especially around mechanism and understanding better of why are we seeing such a higher accumulation, and be able to look at more of that causation effect as well. And then just to kind of show you some of the other plastic materials that we were talking about is we're also seeing consistent trends showing increase in our normal brains for all of our major polymers. So, I mean, we're really highlighting polypropylene here, but we also see increases with polyvinyl chloride, styrene, butadiene, rubber, as well as overall total plastic material, and seeing an overall increase throughout all of those as well. Another thing we wanted to do is, you know, it's one thing to be able to say that there is these plastics, but in order to say that, we also need to be able to visualize these things. So from taking those tissues and digesting them down and being able to palletize these, we've also continued to really work to advance the technique in terms of visualization. So we have an amazing scientist with Dr. Eliane El Hayek, who works in our group, and she did some great TEM images to look at the particles that we're seeing within the brain. And as we can see, these are very shard-like and a roughly consistency around 175 to 200 nanometers in size. So this led to our newest study, is because as you saw in that previous image, a lot of these plastics tend to agglomerate together, so really want to stick together. So this is where we worked with Aaron Ederle at what was formerly NIOSH. So, you know, great scientist, and really been able to work with him to be able to better disperse these particles, to be able to get a better idea of what the size, shape, as well as overall length and width of these. And what was a very interesting finding is that within the brain itself, the length of them is roughly around 175 nanometers by about 45 to 48 nanometers in width. And then the kidney and liver were a little bit smaller overall in size, but very consistent in terms of the shape that they had. And even looking at the distribution of the lengths of these throughout all the different tissues, we really didn't see anything over about 500 nanometers, with maybe like 1 or 2 outliers that are roughly within the 700 nanometer size. So really wanting to emphasize how it's important to see what's biologically relevant, and showing that what we're seeing in the body is more on the nano size versus what we're seeing in micro scale is what we're seeing in the environment. And then just to additionally confirm, with these agglomerates, we did do some Raman spectroscopy to be able to look at these. And if you look at in orange, that's actually going to be a analysis of just a high density polyethylene structure. And then the one in black is actually going to be what we analyze. So these are our actual samples. And we see that these line up very nicely Only difference is we do have this carbonyl group that is forming, but that is due to just how old these plastics are, and just because of UV oxidation, that's where we're starting to see a lot of these carbonyl groups as well.
Okay.
So another visualization technique, and I know I don't have that much time, so I'll kind of go a little bit faster through these.
Thank you.
But a new technique that we've been establishing is the utilization of polarization wave microscopy. So pathologists like Rama Gulapalli can use this technology to be able to look at just a traditional H&E slide. But then from there, whenever we incorporate this new technology, we start to see where a lot of these inclusions of these plastic materials start to form. And even within the kidney itself, you see this structure here, that's actually the glomerulus, or what helps filter the blood. So you see a lot of that there, as well as you see this kind of line of these plastic materials. This is actually going to be the distal tubule, so actually where a lot of these plastics are being removed from the environment, or from our bodies. And then within the liver itself, we're also starting, we're showing that we're seeing most of the accumulation within the liver lipid as well. And again, going back to some of the points of where plastics do start to, or like to follow lipid, this is one of those confirmations that we've been doing as well. And then we just did some additional confirmation to show those shards as well are more carbon-based than potential other materials like cholesterol or others. And then whenever we look at the brain, we are seeing like some of those materials are localizing within the vascular wall, and specifically if we're looking here within the vascular wall, as well as within the immune cells of the brain itself as well. And then just to do some additional confirmation, like we're doing, just to test, are these actually of polymer consistency? This is actually what the particles would look like normally, and then we expose them to chloroform. And then after 24 hours, you start to see them forming together. And then we heated those to about 130 degrees centigrade, which is roughly the melting point of polyethylene. So you start to really see how those tend to agglomerate together. Now, to just finalize some of the areas that we're moving towards now is within that study that was looking more specific at the frontal cortex. Now I was able to collect a whole hemisphere and really working to do a mapping of the entire brain to be able to identify what regions have more plastic than the other. And that'll help us start to answer a lot more questions around like potential neurodegenerative disorders or other conditions as well. So with that study, what we did is we actually did coronal sectioning of the entire hemisphere, and then we collected both gray matter and white matter. Now, I will say that this is an N of 1, again, just because human tissue are very hard to come by, but I mean, we are working to continue to look at that moving forward as well. But then whenever we look at overall concentrations, one thing to really highlight is that all plastic concentration in the white matter is significantly higher than what we're seeing in gray matter. And that also falls in line through all the different plastic materials that we're also able to identify as well. And then just to kind of give you an idea, this is an overall distribution of the entire hemisphere currently as it is. And we are starting to really sparse out a lot of this information to be able to say, like, are we seeing more in hippocampus, lateral geniculate nuclei, dura mater, as well as looking at the different types of nerves, so optic, olfactory, even just the arteries within the brain as well, to be able to identify a lot of these things. So just in conclusion, micro nanoplastics are ubiquitous within our environment. I mean, we're the first people to really show that these are going to be nanoshard-like, which hasn't been described in the literature as of recently, but really understanding what those nanoshards do and how they enter the brain, the retention pattern is going to be very important. But really advancing our techniques of quantification, identification within biological tissues to be able to answer the questions around distribution and potential health impacts is very important to what we're really trying to do moving forward. And with that, I just really want to highlight all the amazing people from my mentor, Matthew Campen, to all of my collaborators, all the students that work with us, a lot of our funding resources. And really this work can't be done without any of them. And with that, I just want to say thank you guys very much. And I believe we can open for questions.
Thank you so much, Dr. Garcia. That was really informative. And the takeaway for me is the word nanoshards. which I think we will be hearing more of. So the chat is now open for questions. We have a couple questions that came in from people prior to us opening this. Let me just share these. The first one we kind of covered, but I want to repeat this. The first question is there any evidence that older people are at a higher risk for microplastics in the brain, specifically things like earlier onset dementia or other effects on senior health?
So in terms of that, we actually did look at age relation, and there really wasn't a correlation with age specific. So it's more of the environmental fact that since there is more plastics present in 2025 as compared to what there was like in 2016, that's going to be the main factor with everything, at least from what our preliminary data has shown. So also too showing that there may be some type of clearance mechanisms that we still need to uncover, but ultimately age relation wasn't showing a trend at the moment.
Got it. And Dr. Garcia, you mentioned you spent some time in Hawaii. So here's a second question. I live in Hawaii and was wondering if plastics have been detected in rainwater in remote places like Hawaii.
So I can't answer that question personally. I mean, I'm sure that there's been a lot of studies out, especially focused on more the environmental area. As far as I'm aware, with what we've done is more looking at plastics within the soils as well as along the beaches to be able to analyze. But I think that is a very important question, and I mean could also be playing a role in this bioaccumulate or this overall accumulation in our environment as well as our food sources.
Great. Next question. What recommended actions can we take to change the containers used in the products used for containers? And I think you could just say packaging.
Right.
Yeah.
This is an area where I guess it is sort of a dual responsibility. You know, sometimes I think we really have to hold the manufacturers and folks responsible, because unfortunately consumers are sort of in a very interesting situation where they sort of have to purchase products that come in endowed with certain types of plastics. So it's almost impossible to avoid them, right? So there's a huge responsibility amongst manufacturers. And as you said before, change in culture and making this sort of incentivizing manufacturers to use alternate materials. But then again, there's also a personal responsibility where you could take the step, if you could, and it's economically feasible, to avoid some of these exposures by eschewing purchase of products or food that contain these plastic materials, right? Eating fresh vegetables and not. So those are things that I tell my patients as well, but there are common sense things that you could do within your control, so you don't have to feel as if you're trapped and it's not possible for you to change. There are things that you can take personal responsibility for.
Thanks. Yeah. And in our overtime section, we're going to talk about systemic change and changing laws, so it's easier for people to avoid plastics. So here's another question. Is the body able to eliminate plastics once it's in our bodies? And I think I know part of the answer, but and how does that happen? And you can say the word poop. This is a family webinar.
Yeah. So in terms of that, what we've seen is I think one of the definitive points is we're still learning around the mechanism of how they're actually being absorbed through the GI tract. Now, a lot of the plastic materials that we're consuming are going to be both on the nano scale as well as the micro and even macro scale. So more visual and things along those lines. So those will definitely be eliminated and excreted out of the body. And I think in a lot of ways are just even from our recent data that I just showed you too, like our body does have a way of eliminating these things. Now, at some point, it just depends on how much it's able to eliminate and what part of the body. So that's why it's important for us to really look at the overall biodistribution. But that's kind of where we're at right now. And Sanjay, if you want to add anything, and especially from the cardiovascular perspective, I think that's very important as well.
You said it well.
And what is this is a hard one. What is the largest source of plastics ingestion for the average person?
It's a good question. I think it's hard to say precisely, but I think it depends on the habits of the individual too, right? So certainly, I think there is a component from foods and foods stored in plastic materials. And again, it depends on, as I mentioned to you, there's also penetration of all of these natural ecosystems. So everything from honey to green foods now increasingly are demonstrating evidence of plastics, right? So I think food is a big component. Water, certainly, I think some of the other infrastructural, components, pipes, etc., and some more evidence that those could also be sources of ubiquitous plastics. There's also, there was a paper, as I mentioned, in Nature published recently, where airborne plastics are absorbed by plant leaves, right? So there's a component that interrelational plastic exposure certainly occurs. I think a lot of particulate material, I think, is endowed with nanoplastics as well. The exact contribution of that to cumulative exposures is not currently known.
Great. So we have a final question, which we're actually going to cover in overtime. I did not write this question. Someone typed in, please talk about extended producer responsibility and legislation like that to hold companies accountable. So we'll get to that in a second. Andrew, could you please put up our final slide? I want to thank all of our presenters this evening. This has been very, very informative. And I want to mention that Beyond Plastics does educational webinars about 4 times a year. We're not a webinar mill. We just schedule one when we think there's a very timely, important topic that people want to hear about. And I want to urge you all to support the work of Beyond Plastics. We are a nonprofit. You can go to our website, beyondplastics.org, or you can take your phone and click on that QR code right now. I know there are so many important issues on our mind facing our country and our world, and a lot of demands on your financial and philanthropic resources. So just put us in your top 3. We don't need to be the first group that you donate to, given what's happening around us. But please consider giving so we can continue to do this kind of work. So I want to thank all of our presenters and give you permission to leave and go get dinner. And then particularly New Yorkers, if you want to stay on, we'll just spend a couple minutes talking about the latest exciting news related to the Packaging Reduction and Recycling Infrastructure Act, which is pending in the New York State Legislature, and if adopted, will be the most comprehensive packaging reduction law in the country. So thanks, everyone. Good night, and stay with us if you're a New Yorker.
Thanks, Judy. It's great to meet you. Kathy, such a pleasure. Vanessa, great to meet you. Marcus, good seeing you.
Good seeing you too, Sanjay, and thank you guys very much. And you guys all have a beautiful and great evening.
You too. Thank you.